Okay, the clock just tipped over to 3 o'clock. It's time to welcome you to the 10th Geography Department Colloquium of the fall semester. This was scheduled to be the 11th, but Tropical Storm Ed derailed that, canceled last week's, and Guashan Yang will present her colloquium on December 3rd. Okay, that's the announcement for the day. Today we'll bring you Dr. Alisa Coffin of the U.S. Department of Agriculture, Agricultural Research Service, working in the Southeast Watershed Research Laboratory in Tifton, Georgia. She's also adjunct faculty in the College of Agricultural and Environmental Sciences at the University of Georgia. The title of her talk is Geographical Applications in Agro-Ecological Research for the U.S. from Point to Region and Measure to Model. Now, Alisa has a BS degree in environmental studies from Rutgers University. As a side note, she actually created her own major curriculum in sustainable agriculture and spent time at the Land Institute in Kansas. In some ways, where she is now is coming back home to where she started. Then she went to the Graduate School of Design at Harvard University, where she earned a master's in landscape architecture. Then after a few years after the graduation from Harvard, she came to the University of Florida and actually is a PhD graduate of our program. I was fortunate enough to be her advisor. And I say fortunate because she did it all on her own with the help of her family. I didn't do very much at all. This was her dissertation. Now, Alisa coming here is interesting because it's good for current graduate students and faculty to see where our people have gone after graduation. Many of our PhD graduates go on to academia, but others don't. Some go to NGOs, some go to government agencies in various capacities, and a few go into private industry. One of the department's claims to fame is that nearly all of our PhD grads are doing work that they want to be doing. But many of the people did not have a nice linear career progression. There are many alternatives, and it would be useful and interesting for the grad students and the faculty to hear a story of one of the successful alternatives. And I've asked Dr. Coffin to tell us a little bit about her path, mostly to focus on her current research, but we'd like to hear a little bit about how you got where you are. So the floor is yours.
Well, thank you very much, Mike. That was a really nice introduction, and I'm going to go ahead and share my screen now so that will go away and share and start my presentation. There we go. Y'all see that? Mm-hmm. Okay, great. All right, well, thank you very much, Mike, for the introduction, and it's a real pleasure to be able to speak here today in this colloquium. I remember going to the colloquium when I was a student and giving my colloquium talk when I was a student, so it's really nice to kind of come back home to this and have the opportunity to talk about the work that I'm doing here at the USDA. So I'm going to go ahead and just get right into this. My thing will go forward. So just a broad brush of what's going to happen in this talk. And I told Mike earlier, I'm kind of used to giving talks now in 15-minute increments. So it's been a while since I put together a talk for a period this long, but I'll try to do my best to stick to the time frame. So I'm just going to talk about introduce myself, talk a little bit about my background. I'll talk about the research context where I work, both within the USDA-ARS Southeast Watershed Research Lab and also within the USDA Long-Term Agro-Ecosystem Research Network, which my location is a part of. I'll talk about some general research objectives and specific research, and then some of the current and future efforts that are going on. So these are all my little icons down here at the bottom. I started out at Rutgers University and Cook College. As Mike said, there was no such thing as an agricultural ecology degree at the time. So I became inspired after I went to my second year ecology class as a sophomore and heard Dr. David Ehrenfeld, who's a well-known conservation biologist, talk about human ecosystems and agriculture. And he mentioned the Land Institute, which, kind of captured my attention. And I was kind of at the point in my college life where I really needed a break, and I wanted to get out of New Jersey. Sorry, New Jersey, and anybody from New Jersey, but I really needed to get away from New Jersey. And so I saw the opportunity to maybe go and take a semester off. And so I did. And it was a really kind of opportune moment at the Land Institute where they were going through some transitions to a different program. But I got to go there and have a really intensive kind of educational program there, learning from Wes Jackson, who's a really well-known figure in sustainable agriculture, and Wendell Berry and others there. So it was a highly influential period of my life, and kind of inspired me when I got back to Rutgers to take on this program of trying to put together courses in ecology and agriculture. And it wasn't so easy at the time because the way Rutgers is organized, we had different colleges, and agriculture was in one college and ecology was in another college, and they were like 15 miles away from each other, and I had to take buses from one class to the other. It was quite an undertaking, but I managed to make it work. And I also learned there about landscape ecology, which kind of fired my thinking about what to do after my undergraduate career. I ended up going to grad school at Harvard to study landscape architecture and landscape ecology, where I met Mike Binford there and Richard Foreman, who taught the landscape ecology courses. And then after that, spent some time working in the landscape architecture field, and then went on to the University of Florida for my PhD, where I really sort of drilled down into the issues of road networks and road ecology. As far as my experience goes, I have lived in several different places in the country, apart from New Jersey. I'm originally from Washington, D.C., but I've lived in Kansas, Boston, Florida, Colorado, and Georgia. I've also lived overseas for a while. Between my undergraduate and graduate career, I worked in Israel for a couple of years. And then immediately after my Harvard program, I got an award for a Fulbright fellowship and went to Bolivia and started working there with some of the colleagues that Mike's been working with from the University of Chicago to look at socio-ecological patterns of land use in the Bolivian Altiplano. And then from there, I went on to an academic appointment at Ball State University for a few years. And at that point, I decided that I really loved research and I wanted to get back into it. And so I quit teaching and I went to graduate school, and it was kind of a long story, but took a while to get through with having kids along the way. But ended up coming out of that with a postdoc at the USGS, and that eventually led to some consulting work where I lived in Colorado, and then finally to this position where I am today at the USDA-ARS in Tifton, Georgia. So that's my sort of long and tortuous path through my career, but I would say it's been an interesting ride. That's for sure. So I'm going to talk to you today a little bit more about the work that I've been doing, give you a little bit about the context, both locally here in Tifton and nationally in the programs that I've been involved in. As I mentioned, I work at the Southeast Watershed Research Lab, which is the home of the Little River Experimental Watershed, or the LRU, we call it. So the watershed was authorized by Congress in 1959 in a program that was a national program for experimental watersheds. After a few years of research to kind of identify exactly where the watershed would be and where its headquarters would be, they settled on this location here in Tifton, which is also the Coastal Plain Experiment Station associated with the University of Georgia. So this map here on the slide here, you can see the outline of the watershed. It's a series of nested subbasins. There are 8 of them, and in this watershed, we've been monitoring stream flow and water quality for around 50 years. We also have been monitoring land cover, doing our own land cover surveys for at least 20 years. We have a network of precipitation and basically climate stations throughout the watershed. And in the last 5 years, we've also been establishing some flux tower locations as well in some of the cropping areas that we're researching. This lab has over the years produced a lot of valuable research for the USDA, including critical research on vegetative buffers that have influenced NRCS recommendations. It's quite actually astonishing to me to learn all the time about new things that I didn't realize the ARS did that have fundamentally affected our economy. So, for example, permanent pressed cotton, that came out of an ARS lab. And blood thinners. Blood thinners were produced as early on as rat poison that was produced in an ARS lab. So there are a lot of things that the USDA-ARS have done over the decades that kind of, have really fundamentally, influenced areas of our economy. And so this is one of the places that has done that by doing this sort of fundamental research on riparian buffers. Oops, sorry, one too far. There we go. So our mission here at the watershed is, hold on, I gotta move this out of the way, is to develop a scientific understanding and associated technologies of watershed systems that are essential to maintaining and enhancing the environmental and natural resource base upon which a viable, sustainable, and productive agricultural economy depends. Our unit here employs 8 scientists. We have 2 more that are, we're in the process of hiring actually, and then we have support staff. So, and that's grown a lot. When I came here, we were 5 scientists, and now, we've merged with another unit, and we're also hiring. So we're expanding our base. We are a multidisciplinary group consisting of an ecologist / geographer, a chemist, an agricultural engineer, a couple of hydrologists, and we're hiring a modeler. And also, we've just incorporated 2 entomologists, and a weed scientist is coming. So we have a really kind of interdisciplinary group, and it's a really nice group to work with. We all work together pretty well, and we try to support each other and publish together. So that's kind of fun. So we have a soils lab, a chemistry lab, insect labs, a water quality lab, and my lab, which is the remote sensing and GIS lab. We also have soil and plant processing facilities. I mentioned we're co-located at the Coastal Plain Experiment Station. You can see an aerial shot of some of our buildings there. And then we have lots of field sites. Of course, we have all of the stream gauge stations and weirs throughout the watershed. We also have some sites where we do plot and field level studies. So in this research that we're doing here, we spend a lot of time actually working in the field, gathering, measuring data at point levels and plot levels. Our funding comes from Congress through the USDA, and it's funneled through these sort of national programs. And increasingly, these national programs are looking to, pushing us basically as a scientist to explore cross-fertilization across these programs. So we may be funded by one program, but we're also collaborating with people from other programs. So we have 4 different funding programs within my unit. And, if someone from the NP216 project needs me to do some geospatial analysis, then I do it, you know, and we publish together. So that's really been a really nice and interesting part of this job. We're also part of the long-term agro-ecosystem research network, which I'm going to describe in a minute, and part of the conservation effects assessment program, which has been a long-running program to look at the effects of conservation practices in agriculture. And we have a number of research collaborations that are ongoing, funded by NASA, NRCS, NIFA, and other university partners. So the point about this is that when we do our research, in order to be able to say anything about what's happening at broader scales, which is really our mission, all of those things have to rely on some sort of a quantitative scaling process to be able to make those assessments. And so we have to extrapolate or scale our measurements from point levels to plot levels to field levels and to landscape levels. And some of these things we are collecting. So you see the things that I've gotten here in white fonts are measurements that we're actually collecting. And then we have to also do some spatial analysis to be able to extrapolate or model results at higher, broader areas. So this is an example here on how we do some of this work. On the left is a map of one of the farms where we work. That grid of black dots that you see there are all the points where we go out and collect hand samples of biomass and soils. The red triangles are, this is a field map that I had produced from 2019 that showed where we had some intensive sample points where I had soil moisture sensors located, and we did some intensive sampling. We also fly UAVs here. I have a UAV program that I started after I came here. This is one of our UAVs up there collecting some data over one of our research farms. So I also mentioned the long-term agro-ecosystem research network, and our unit is one of 18 sites in the U.S. We are referred to as the Gulf Atlantic Coastal Plain. So you can see us over here in the southern part of Georgia, and there are a number of other sites across the country. We collaborate a lot. I work a lot with Hillary Swain at Archbold Biological Station. It's actually the Buck Island Ranch where we've been working, and the ONA, UF-ONA facility there as well. But the LTAR network started in 2012. The focus is on sustainable intensification of agriculture using this shared research strategy. And the idea is that we have this coordinated agro-ecosystem research that's going on along with an information management program to provide new management technologies, information, new concepts of agricultural systems for our stakeholders. And that these stakeholders in collaboration with us lead to these new partnerships and innovative ideas that then feed back into our science. So we're very much interested in this idea of co-production of science, and then ultimately being able to provide a service to the consumers and the public and land managers and policymakers. We take this tack of looking at agricultural sustainability, kind of relying on the sort of traditional three-legged stool idea, or the sort of three I call them domains of environment, productivity, and rural prosperity, which is a USDA term. We've been kind of more referring to that as human well-being or the human context. The basic idea is that each of these LTAR sites has an experimental design that compares management practices of what constitutes a business as usual system with an aspirational system, so BAU versus ASP. And that the details of those designs would vary from one site to the next, but the point is that we're all sort of working under this framework of what is business as usual versus aspirational. In some places, they have several different aspirational possibilities that they're comparing. The work in the network is accomplished in working groups and at a leadership team level, and I serve in all of those. I'm a member of the executive leadership team, and I also run the remote sensing and GIS working group and another research project that I'm going to talk about here in a minute. So if we wanted to quantify the LTAR network, there are a few ways to do it. There's non-spatial ways that we're all pretty familiar with as researchers, using statistics, valuation of resources and services, measuring interactions, doing clustering analysis and network analysis that don't necessarily spatial components. And then there's the spatial side of things, which as a geographer, this is the part that really, really interests me and the part that I came here to talk to you about. So, and this involves necessary use of things like spatial statistics, interpolation and extrapolation methods, and methods that measure spatial interactions like gravity models and telecoupling and things like that. The basic point is here, and this is what I have to stress with my colleagues, is that what we're trying to do is map values by matching them with attributes of latitude, longitude, and elevation. And this may seem like really basic information for you all as geographers, but I'm telling you, this is not basic information for a lot of people and a lot of scientists that don't have geographic training. So this is the main, I guess, contribution that geographers and spatial scientists make to these kinds of collaborations. It's really bringing this awareness to the table. And as part of that, I've sort of tried to express to my colleagues in the LTAR network, to think about how we work together and the scales of analysis that we use. So we have the individual LTAR nodes that work at the plot level, field level, farm level, enterprise level. And that's fine. And then we may even have some subnetworks of LTAR sites. As I mentioned, I work a lot with the Florida site. So we just put out a paper. We're just about to put out a paper together. We've got it in review right now. And there are other groups that are kind of clusters of subnetworks within the whole LTAR network. And then we also have coordinated LTAR network research going on. And to be able to do this, we need to have plans. We need to have a research plan. So it's good, it's valuable to have a research plan to get yourself organized and working. But you also need to think about cross-site collaborations, and eventually the goal is to get to the point where we can do network-wide research. And as we move in that direction, the importance of spatial data, and especially remotely sensed Earth observation data, becomes essential. And so we need to have kind of these geospatial frameworks to be able to get us to that point. So in thinking about this, we're also thinking about how we sort of turn measurements as indicators into indicators that we can interpret as outcomes at these different scales. And so we have a couple of efforts that are ongoing in the LTAR network that I'm going to sort of review here. But one of them is a sustainable indicators project to identify what those indicators would be. Another one is a project to identify a spatial framework for analysis, which we call a regionalization project. So to get at it, sort of the geographic nature of the problem, we have, at both of these levels that I've mentioned, my sort of unit level and the sort of national network level, there is a geographic problem. We need more food, fiber, and fuel, and we need to reduce the adverse effects of agriculture. And precision agricultural methods for farming, ranching, and conservation are kind of held out as critical pathways for being able to achieve the sustainable intensification of agriculture for all of these realms. But one thing we need to think about is how these ecosystem services trade off at different scales. So we need to understand the environmental variability of the places where we're working, and we need to understand that spatial and temporal problems have to account for scale, uncertainty. We have to be able to manage big data, understand their characteristics, volume, latency, and so on. So spatial analytical frameworks are needed and to be able to accomplish this work. And the rest of this talk is going to kind of just give you a couple of examples of how I've been trying to engage with that over the last few years. So the first sort of effort is this broad sort of national scale network. It's looking at this, we call the LTAR regionalization project. So as a geographer, when I came into this project 5 years ago, I saw this LTAR map, which looked a little bit different than the one that you see here right now. But I looked at the map and I said, why are those boundaries where they are? And no one could really give me the answer. And so I just kind of said, well, we really have to do better than this. We have to understand why those boundaries are where they are, and are they meaningful? And, if I can't explain why Great Basin's boundaries are where they are, and they can't explain why Gulf Atlantic coastal plains boundaries are where they are, we have a problem, because we don't have a common spatial framework that we can use to compare our results. And so we sort of embarked on this project in 2018 to begin looking at spatial frameworks. We started with looking at existing frameworks like ecoregions, major land resource areas, HUC watersheds, and ERS farm resource regions. And we didn't think that any of these were really suitable for the research that's going on in the LTAR network. So we began our research project in the regionalization group to carry out a few different things. One was to do this analysis of boundaries. Another one was to look at the representativeness of the network, and then to look at the representativeness of each location in the network. So for the boundary analysis, what we're trying to do here is to do a sort of clustering and classification of national data sets, testing different clustering methods using unsupervised classification. This shows you an example of not a final product, but just a kind of an interim product that Sarah Gosley put together, just looking at climate and soil variables and dividing them up into 50 different classes and seeing what it would look like. So we're in the process of exploring the data right now, trying to develop a conceptual framework that can support a clustering analysis process. And by that, I mean like a theoretical framework that will tell us what those relevant variables are for each of those domains of production, environment, and human context. And the outcomes of this, we hope, will be a framework that we can use to describe the boundaries, what we call crisp boundaries. But we're also sort of making room there for the possibility that a given pixel may belong to more than one site. So you might, what we might call fuzzy boundaries. And in our effort to do this, we reached out to Bill Hargrove, who's a researcher at the U.S. Forest Service, who works a lot with this type of analysis, did a similar analysis for the NEON network. And we're running this now for the LTAR network to look at the representativeness of a slew of bioclimatic variables and how those relate to the LTAR network. So we're doing this clustering and classification of using local areas of interest from LTAR sites to figure out those clusters and look at how representative the network is. And this is a preliminary result that you can see here. We're still refining this, but one thing that you can see here, for example, here's the Great Salt Lake. So that's red, and that's very low representativeness. So I guess that's good, because we don't want to have, we wouldn't want to be representative of the Great Salt Lake. But, you know, the other thing that you can see here is that we have some gaps. And so we also want to think about, you know, where those gaps are. And then at a local level, you know, this is hopefully going to help us to understand, you know, what our level of confidence is for being able to understand, you know, how far out I can measure, how far out I can infer from the places where I have measurements. So this little purple star in the middle here, and the green blotch there is the Little River Experimental Watershed. And this is our tentative outline right now of our boundary. But is that correct? I don't know. Is that right? I mean, we kind of did that based on the ecoregions for the area, but we really want to have a better way of being able to decide this. And then with that, we should be able to perform better regional characterizations, you know, asking things like, what is the extent of our actual management patterns, you know, like our crop rotational systems and so forth, and how do the ecosystem services vary among scales and locations, and what are the trade-offs and synergies within and among those regions. These are a few graphics that I've picked out. This is the two here, the one showing Florida and the Southeast are from this paper that we're working on with the folks from Archbold, and the sort of spider diagram, I guess, or it is kind of showing how we've decided to depict the trade-offs at these different scales and across these different regions. So again, getting down now and back to the Southeast Watershed Research Lab, you know, this relates to our site because we're one of those LTAR networks, and we have to accomplish that research at our site to be able to share it across the network. Our site is driven, and all ARS locations are driven by these 5-year research plans where we collect field data. We do a lot of analysis, you know, reviewing and harmonizing the data and applying geospatial methods and interpreting the results. And so I'm going to give you an example here of one that we just did. And this is another paper that's under review at PLOS One, looking at responses to environmental variability by herbivorous insects and their natural enemies within a bioenergy crop, which is miscanthus. So in this project, our objective was to investigate insect natural enemy and spider abundance along with environmental variables as factors that shape the within field spatial distribution of herbivorous pests, specifically phloem feeders, over the growing season and within this relatively new crop. So to do so, we compared outcomes from non-spatial linear regression methods with geographically rated regression models. And the idea was to find out, you know, can these spatially explicit models better predict pest-predator environmental dynamics? So you see the location map here of where our sites were. Each of these points was a sample location. And this graphic here on the right kind of shows you the outcome from the geographically weighted regression, which was very pretty, but kind of hard to understand. So we have a farm in Tai, Georgia, which is about 10 miles away from here. And it was planted to miscanthus for several years as perennial grass. We had 81 sample locations that we sampled over 2 years for 3 times every year. We sampled for insects in the ground and in the canopy. You see that here is the pitfall trap that we used for the ground dwelling insects. And then we had sticky cards. They're little yellow cards that stick on a bamboo pole in the crop. And we also measured environmental variables. We took direct measurements of NDVI with a handheld sensor. This was before I had the drone, so we used a handheld sensor for that. And then we also did the same kind of coring of soils, of the top meter of soils, and characterizing the top 15 centimeters of the soil core. And we also have a weather station nearby, and then did a sort of terrain analysis as well. So our results from this project, we did a couple of different things. We did sort of a non-spatial global model, and then we segregated the data into seasons, and then we also did sort of non-spatial models with the seasons, and then we did these geographically weighted regression models as well. And these are some of the results that I'm not going to go through all the very fine details, but basically, had some negative relationships with elevation, silt, and NDVI, and positive relationships with some of the weather patterns. And of course, with the geographically weighted regression, those are all over the map, literally. So you have to take a little more care in interpreting those. And this is how we did it. We summarized the data, and this graphic that you see on the left is, you have to look at it for a little while to really understand it. But basically, up here is the little map of our field that we divided into 4 quadrants. So we had 3 seasons, early, mid, and late, and 4 quadrants, and 7 variables. So each one of these little blocks that you see here represents, here's one quadrant. So here's the elevation variable in the early season, and these are the 4 quadrants. And basically looking at the map here, so here are the coefficients. This is the surface of coefficients here. And then the dots that you see in here are the actual measurements that show, where we took the measurements, and it's also a measure of what we called credence. It's not confidence, it's credence, because it's not really, we're not really sure of the statistical validity of it, but our statistician said, yeah, call it credence. So, okay, we call it credence. So if the coefficient to ratio to the standard error is, highly positive, then we gave it a black triangle. If it was highly negative, we gave it a red triangle. So what you see here is that across the field, you have different relationships. In one part of the field, elevation was highly negative. Another part of the field, it was highly positive. And this, of course, changed over time. So, here's the mid-season, and here's the sort of annotated version of that. And here's the late season, again, the annotated version of that. So, to just kind of tease this apart and find out what the story was really pretty hard. It took us a while to kind of figure this out. But this is how we managed to depict it. And, we were able to make a few conclusions from this. One was that in the seasonal non-spatial models, plant health was a significant predictor of phloem feeder abundance as measured by NDVI. And, in this case, NDVI could be mean that it's a healthy plant, or it's, so it's an attractant to herbivorous insects. And it also could mean that that we're seeing the effects of herbivory in the case of a negative relationship. And that's what we found more often, was a negative relationship with NDVI, although you saw you will see in our graphs that there are some places where we have positive relationships. But we concluded that, especially in these non-spatial models, we saw that phloem feeders preferred the weaker plants. And the geographically weighted regression models provided spatially explicit information about these complex responses to environmental variability. But as I mentioned, they're really kind of hard to interpret because they move, they shift in space and over time. So you have to kind of like take it apart very carefully and look at them over and over until you figure it out. What we found was that the variables with the greatest spatial consistency included NDVI, which was negative across the entire field in the middle of this, in the mid-season, the second period of sampling. and that natural enemies were positive. You know, there was a positive correlation between natural enemies and phloem feeders across most of the field in the early season and the entire field in the mid-season. And the takeaway from that is that unraveling these complex and changing relationships among environmental variables is not simple. It requires this fine-scale local data that we've been spending so much time and energy collecting. And it also requires the use of analytical methods that are spatially explicit. So that training in geography that you all are getting there is really essential to this work. And so we have some things that are happening. You know, there's always stuff happening around here. But we've got, you know, we're planning right now for the next cycle, 5-year research cycle for the NP211 project, which is what pays most of my salary. And the lead scientist for that is Dr. David Bosch. He's going to be retiring in about 18 months. So I'll be taking over from him as the lead scientist on that project. We have a new building that's been authorized by Congress, and we're in the design development stage. So hopefully we can move out of these little, what my husband likes to call trailers. Says I work in a trailer park, so hopefully we'll get a more dignified building. And in the remote sensing and GIS lab that I'm you know, working, that I kind of lead here, we're developing a collaboration with Marguerite Madden at the UGA Center for Geospatial Research and Lynn Seymour at the Department of Statistics in UGA to kind of develop a, you know, a coastal plain center for remote sensing. We have, you know, opportunities that come up all the time. I would like to let you all know that there is a postdoc opportunity currently open in my lab. It's with the ORISE program. It's a postdoc to look at small unmanned aerial systems for remote sensing of agro-ecosystems and high performance computing. So that would sort of put the person into a group of other postdocs that's working with our high performance computing systems called SciNet. And that's about it. I have exhausted my time, but I do have a short video that I want to share with you guys because it's absolutely amazing. And my technician did it, and he is a master of videography with drone stuff. So you guys are going to enjoy this, I hope. Can y'all see that?
Okay, Alisa. Yeah, we have some time for questions, and Alisa, you should know that sometimes students have or people have things going on at 4 o'clock or a little after, and they may leave ahead of time. But I'll stick around for a while and answer questions, so you can answer questions for a while. Anybody up?
I see a couple of applauses there.
Yeah.
Thank you. Thank you.
Yeah, I'll do that too.
Well, the person in my unit who does this amazing drone work is my technician, Kobe Smith. So he gets all the credit for that. He does this also as a side job, doing videos for real estate folks and all.
I see Dr. Whalen has activated his microphone.
I have. I'm ready to speak. I was actually waiting for Tim to speak up because I suspect he's going to ask something very similar. Hi, Alisa. Nice to see you again. Osvaldo and the family. Benny must be pretty big by now.
Yeah.
Geographically weighted regression. And the results you got from that. I mean, did you have any problems with sample size? I mean, I was trying to work out how many traps you had there, and then when you divided it up into quadrants, how many you had left in each quadrant, and when you divided up sometimes each quadrant into quadrants again, I wondered how many points you had in each one of those and how significant an unusual observation might have had.
So the actual quadrant division didn't happen before the regression. So everything was used in the regression. So we had 81 sample points times 6. So it would have been, for the global. But then we had we combined. So we did 2 years of data. So 81 times. So we had 162 sample points in each regression.
Okay, so because I mean my memory,
which is on the low side, I agree. It would have been better to have more, but we were still within the tolerance, I guess, of the method. So
I mean, because with geographically weighted regression, you kind of, you move a spyglass around, right? A weighted spyglass around. And so I wondered just how many in that spyglass, how many observations you usually have.
I would have to go back and look in the paper, but it was always well over 100. So I mean, it's a small, it's a small field. It's not that big. So they were pulling the models were pulling basically from everything within, almost across the entire field, but not the,
Okay.
So it was looking at a lot of the neighbors. Yeah.
And the same sort of issue with boundary effects. I mean, if you moved your spyglass close to the edge, did that?
Yeah, I mean, I did not directly deal with the boundary effects, so that's probably an area that was not addressed in this. So there could be some issues there. That's possible. Yeah.
I'm really just speaking for Tim. That's all.
Go ahead.
I had a related question to that.
He's there.
Sure.
Geographically weighted regression in terms of, so you're using in terms of the kernel, you were using a fixed, or was it an adaptive kernel? I guess, I see what you had problems at the boundary, but was it a fixed or adaptive method?
It's an adaptive. It does an iteration through the so it finds the best fit for the kernel, and it's something called a golden search routine that ArcGIS includes now in their ArcPro version.
Okay.
So one thing that is, I guess, a downfall of this geographically weighted regression method is that it is specific to the software, and they're pretty upfront about that in their description of it too. But they use what they refer to as a golden search routine. So it is.
My problem with GWR has always been, how do you validate the models? Because you'd have to, if you put points for a data set that you didn't use in the estimation process, how well is it at replicating, and those points? How well is it? The problem is we don't have the standard errors. We have to you have to calculate those, and there's a lot of issues with GWR. It always does better because it only looks at localized values, and we know things that are close together are usually similar. So of course it always does better than traditional regression. But the thing about model validation, there's questions that come into play. So, thank you.
Tim, what would you recommend as far as like being able to overcome that issue, like for validation?
If you have a holdout sample that you can have, and then create, then you could do perhaps estimate surfaces of the beta coefficients associated with each of the variables that came in significant. And then run, run some tests and see from the holdout sample how well the model does.
Gotcha.
And that's one, I think. But I think Dr. Whalen's concerned over boundary values issues with smaller sample sizes, because it does get compressed as you get closer, towards a boundary. There's nothing beyond that. The other thing I can do with boundary effects and to satisfy or to correct for a boundary effect is to actually have a mirror image of the data on the other side of the boundary.
And
that's one way to correct for it. So you could try to, try that.
Thanks. Those are really great ideas. Those are really great ideas. I appreciate that.
Excellent stuff. I was really fascinated. I just had one other quick question. Has anybody looking at, when you got to the classification of boundaries, and you got into the issue of fuzzy boundaries, I could see that. Has anybody looked at how the boundaries perhaps have, even if they end up being predicted, are changing? Do you have a longitudinal data set? We have enough time series data to look at how the boundaries not only are being defined and how accurate they are, what they might look like, but are they shrinking? Are they expanding in different areas due to perhaps climate change and the things that have been experienced over the last 2 decades. That's
yeah, sure. I mean, that's definitely part of the scope of our thinking on this. We want to just try to figure out the optimal methods to begin with, but certainly looking at the sort of modeling future scenarios for climate and the variables that we're using for the clustering is part of it. Absolutely.
That's cool work. That's going to be really neat, because it'll be neat to see how even the fuzzy boundaries, or because there is no such thing as a hard boundary.
No, I know. Yeah, it's all an artifact, right?
Yeah, right, exactly. But it'll be interesting to see how where the fuzzy lines are, where the density of those things, if they're moving in one direction or another, and if that's consistent with other types of things we see in terms of climate change, like changing rainfall patterns or whatever, and changing NDVI signatures and things like that. So really great work. And I again, have a happy Thanksgiving to you and yours. You're doing amazing stuff. Keep it up.
Thank you very much, Tim.
Well, Dr. Whalen and Dr. Fick are the usual suspects for asking questions. Anybody else out there? A couple of grad students need to ask some questions. Well, I have a question, and that it kind of relates to what Tim just asked or just suggested. Is, climate and soils and vegetation are the basis of ecoregions, and even though the ecoregion maps now were actually created back in the 80s and maybe even into the 90s, and things have changed a little bit. What did they miss that made it inadequate for your regionalization and your boundary study?
Well, the big missing factor is socioeconomic variables.
Yeah.
We need the human dimension, absolutely. And this is a really underdeveloped piece of the pie, basically. So that's a huge part of it. And as over the last 2 years, LTAR invested $900,000 to hire a cadre of postdocs who are now working on that issue. Wow. So I have a postdoc, and she's working, she's sort of leading the charge and trying to understand, what are the relevant variables that would be able to contribute to, socioeconomic variables that could contribute to this kind of an analysis. And then the other part of it is the actual production, because we do have the underlying sort of eco-regional, biophysical characteristics pretty well figured out, but the next layer on that is management and what people do with the land, and what are the outcomes of that, and is there a way to cluster those outcomes and characterize those as regions? That's something that's also kind of not very well understood, and it is kind of a false, I guess, classification to say that there are 3 different domains, because they do overlap, but we have to try to simplify to some extent. So that's how we're doing it anyway. So.
You do remember that all classifications are convenient fictions anyway.
It's true. It's true, and we're trying to just figure out what's the most truthful, convenient fiction.
The most useful.
Yeah.
Okay. If nobody else is going to jump in, I have one other one. You did a representativeness analysis of your sites and the regions that they're in. What were the variables? What were the bases of the representativeness? So
that, like I mentioned, that was an interim product that I showed you, but this was based on a clustering of 15 bioclimatic variables. So I'd have to go back and look at our list, but they include soils, climate, I want to say soil carbon. I can't think of the whole list off the top of my head, but it was essentially a similar analysis that they did for the neon domains. Yeah. If you're familiar with that study.
Yeah, quite. I'm still working a little bit for National Science Foundation, and I work in the NEON program, the NEON oversight program. So I see every day what's going on with NEON.
Yeah, So one of the things we're trying to do with that is to be able to do a sort of comparison between LTAR and NEON and see, how the networks are how they compare and how they're different. So.
Right. Okay, it's 4 o'clock, and this is our usual sign-off time. But again, I'll stick around if somebody wants to ask any more questions, or talk to Alisa about just anything else she's willing to stick around for.
Well, I do have to get to another meeting in 10 minutes, but I can stay around for a few more.
Okay. Well, thank you very, very much, Alisa. For those of you who are left, remember the next week we'll not have a colloquium, and students, don't forget to turn in your evaluations. This is how we keep track of who attends. So thank you very much, Alisa. This has been just absolutely wonderful. And Tim's right. This is some fabulous stuff. And I'm really
I really want to thank you and thank everyone who attended, and also thanks for your questions. I really